Chapter 5. Structure and Function of Plasma Membranes. A cell can only be defined with a border that separates the inside from the outside. This border is called the plasma membrane. The selectively permeable membrane dictates what enters and exits the cell. It keeps organelles and ribosomes in, absorbs nutrients and oxygen, and expulses carbon dioxide and other waste. All cells have a plasma membrane and cytoplasm. The plasma membrane is a lipid barrier that separates the outside of the cell from the inside and dictates what enters and what leaves the cell. The bulk of the plasma membrane is made up of phospholipids. These molecules are made of hydrophilic phosphate and glycerol head and two hydrophobic fatty acid chains. It forms a bilayer allowing easy passage of ions, water, oxygen to pass inwards, and carbon dioxide, ammonia, and water to pass outwards. Embedded in the plasma membrane are several proteins with varying functions. Some allow nonpolar, non-charged, or large molecules to pass through, as in protein channels. Others act as enzymes, and others function in communication. Cholesterol helps to maintain some structure to the plasma membrane, since phospholipids alone have the consistency of plant oils. Glycolipids and glycoproteins are found along the surface to help with communication and attachment between cells of the same tissue. Finally, it's important to note that the fluidity of the plasma membrane. First, it is rigid enough to burst open if too much water enters the cell. However, it's fluid enough to self-repair if an object penetrates the bilayer. We'll start with phospholipids. Phospholipids make up the majority of the plasma membrane. Being amphiphilic, they contain both hydrophobic and hydrophilic components. Hydrophilic heads are made up of glycerol and a phosphate. These are able to come in contact with the aqueous fluid of both the cytoplasm on the inside of the cell and the extracellular fluid on the outside of the cell. That's because phosphate is charged and polar, which is similar to water. Hydrophobic tails are made up of saturated and unsaturated fatty acid chains, which are uncharged and nonpolar. Therefore, they don't mix with water. This forms a kink in the unsaturated fatty acid, making these semi-permeable, meaning that some molecules, especially nonpolar, small, and uncharged molecules, can pass through easily. Temperatures can affect the permeability of the membrane as membranes start to shrink in cooler conditions, making them less permeable and easily ruptured. This molecular structure is important as they form a bilayer with water on both sides, therefore creating a cell membrane. The next most common component of the plasma membrane are proteins. They come in two forms. Integrins, or integral proteins, span the entire length of the plasma membrane. Peripheral proteins are found on one side of the plasma membrane, either exterior or interior, and are attached to they could be attached to an integral protein or to a phospholipid. And these proteins have various functions. Some simply help maintain the integrity of the plasma membrane, others act as enzymes, and others can act for communication, as well as allowing certain molecules into and out of the cell. Cholesterol plays an important role in strengthening the cell membrane in animal cells, since no cell walls exist in them. Without cholesterol, the membrane could be easily damaged. Glycocalyx is made up of glycoproteins and glycolipids. These are hydrophilic and help the cell interact with the watery environment and are only found on the exterior side. It also aids in cell identification, determining if the cell is part of the self of the organism or it's foreign, and causes attachment points between cells of multicellular organisms. Glycoproteins are carbohydrate chains that form on proteins, and glycolipids are carbohydrate chains that form on phospholipids. In our next video, we're going to be looking at a very important process called passive transport.